HIV and AIDS, and hopefully it should be lots of really good scientific, biomedical content with some really good interview stuff as well. So today we have Ali and Al Hassan both going to Oxford. I'll let them introduce themselves as well, and then hopefully we can get started in a couple of minutes. And if we can try and keep all kind of sciencey questions to the end as much as possible so the experts can have a go at answering them. Any other questions, feel free to put them in the chat as you go along.
And by the way, this is like a very interactive slideshow, so I want you guys to sort of raise your hand, turn your speaker on if you can, because we'll have a bunch of interview questions and we want like engagement as much as possible. So be prepared to sort of answer any questions.
I completely agree. Yeah. I might not be able to present anything though if I can't get my notes onto my laptop. Have I got them?
Should we start?
Yeah, go on then.
Okay, so hi guys, my name is Adi, and I'll be starting medicine at Oxford in October, hopefully. I did chemistry, biology, maths, and further maths. And yeah, I'm here with
Uh,
Al.
Yes, hello, I'm Al Hassan. I'm also going to be starting at Oxford in October at St. Peter's College. I achieved offers from Imperial, Bristol, and Newcastle, and I did biology, chemistry, and maths over the last 2 years.
Yeah, so I'll talk today about like sort of how HIV in different many ways. So sorry. It's centered around HIV. So the first section will be about physiology and how sort of HIV is spread around the body and how our body deals with it. And then the second section will be about treatment and what current treatments are, how doctors sort of diagnose it and monitor it, and what treatments could be developed in the future and what research is focused on right now. And then the third section is about epidemiology and who is most affected by HIV, what's the mortality rate, sort of all the statistics behind that. So to begin with, I want you guys to I want someone to volunteer and be very brave and say sort of what they know from A level about the structures that are labeled here. If someone can turn on the mic or raise a hand.
I'm trying to work out how to open the chat because that might be a good idea. Just say, oh, what's X, and then they can just put in the chat what they think it is. If that sounds good.
If you can't see the chat, I can also let you know what's in it because I've got it up. Got it?
I can see it as well.
Okay, perfect then.
Oh, I can get up here. Yeah.
Yeah, so yeah, attachment protein is X. So I saw someone say viral DNA RNA. So if we look here that and notice that this is a reverse transcriptase, does that give us a hint to whether this is DNA or RNA? Yeah, it is RNA. So RNA, so you might be familiar with transcription, which is turning DNA into mRNA, but then reverse transcription is turning RNA to DNA. So reverse transcriptase is the enzyme that does that. So it only binds to RNA. And we know that HIV is a retrovirus, like someone said in the chat. So that means that it injects this RNA and then the RNA is turned into DNA. So yeah, what other proteins do you guys think that HIV could have? If sort of I give you a hint that you need to integrate the HIV DNA into the genome, because when it turns into RNA, when the RNA is turned into DNA by reverse transcriptase, it needs to be incorporated into the genome of the host. So I'm seeing ligase. That's a very good guess. It's a
I'll give
very
you a clue. What do most enzymes end in, including ligase? Helicase as well. What do they end in?
like the suffix.
Ase.
Yeah,
Yeah.
yeah. So if we're integrating the HIV DNA, that we call the enzyme that does that HIV integrase. So that
Integrase.
integrates into the host genome. So we'll be going into more detail about how that works as well. And another protein that is seen in HIV is protease. So that sort of helps package the protein, package the HIV virus, and like after it's replicated, so that it can be a fully functional virus.
Am I taking this bit?
Yeah.
Yeah. Okay, so from your current knowledge from A level, maybe even GCSE, could you put into the chat some ways that you think that HIV is spread? Bodily fluids, exchange of bodily, sharing needles, bodily fluids. Brilliant. So that is the main way bodily fluids are transferred, and then we'll talk about what happens with those bodily fluids and how the HIV goes from them into them. Breast milk is included. That's brilliant. Direct contact, sharing needles. Direct contact is a bit vague, but I suppose so, yeah. Okay, so would anyone like to give me the first step of HIV replication, or where it takes place? We won't go through all the steps, but just where it takes place. Hopefully you've done this in AS biology, but when I was talking to two of people who did the same biology spec as me, it depends on how they teach it, but you might know if you've done immunity already. Not quite somatic cells, it might be that. T helper cells, brilliant. So that is the main way that you're taught in biology, that they replicate in T helper cells. So yes, you need a host for the virus to replicate. Viruses aren't live, they cannot replicate on their own. They're acellular, non-living. So yeah. What is the difference between HIV and AIDS? HIV is the virus, AIDS the symptoms. Kind of. HIV develops because of HIV. Sorry, quick question. Is this Northern Ireland spec AS or A2 bio? I'm not sure. If actually you would like to quickly Google that and find out, that might be great. What can happen if you develop AIDS? But yeah, AIDS develops because of HIV. AIDS is a progressive version of HIV with a lower survival rate. Brilliant. Just to clarify as well, HIV is the virus which infects you and causes an infection. AIDS is acquired immunodeficiency syndrome. It's a group of the symptoms and stuff which develop at the end stage of HIV infections. But what can happen if you develop AIDS? What ultimately kills you? If you have less T cells, infection, shut down of the immune system, brilliant, weakened immune system. That's the main thing, which then leads to, as someone said, I think infections. Yes, infections, brilliant. So these can include tuberculosis, hepatitis, which we'll go into in more detail as well. What is the name? This is the most rogue one, which you might not have been taught, of the main treatment for HIV at the moment. Pneumonia is a big one as well. Antiretroviral drugs, brilliant. So we can move on from that.
Oh.
Brilliant. So what I'll be talking through basically is how do we get from being infected with HIV to how does the HIV replicate and how does that lead to the end stages of the disease. So these are the three transmission routes, which I'll go through. So the first one, which is probably the one that most people have heard of, sexual transmission, and the main route for these infections that aren't through the blood is something called mucosal tissue. And it covers a lot of epidermal tissue in lots of areas for your body, and it is involved in your non-specific immune response. You might have also heard it called innate immune response. And it may create mucus, which protects your body from different pathogens and different debris and stuff. It captures them. And it also has a host of immune cells to protect your body. The main one I want you to think about is dendritic cells, which you might not have heard of. And their purpose is to act as antigen presenting cells. So what is an antigen? Foreign protein that triggers an immune response. Brilliant. So they act as antigen presenting cells. So when they become infected with a pathogen, they then take it to your lymphatic system to expose it to other immune cells, which produce an immune response. Okay. So this is a diagram, kind of how that works with other cells as well. So you don't just have dendritic cells, you also have T cells and something called Langerhans cells. And the GP120, which is a kind of attachment protein on the HIV virus, attaches to these and then they move to secondary lymphatic organs. And when within the mucosa and stuff, when this process happens, the main purpose here is, I suppose, to build up a reservoir of HIV infected cells. So we'll go on why it having lots of HIV infected cells progresses the disease. But yeah, okay. Stop. So, the first question, I suppose, now that we're talking about infection and diagnosis, if I am a doctor or you're a doctor and a patient comes in and they have a HIV infection, what would I consider um when I am about to break the news of this diagnosis? This is an interview style question. So
sorry to interrupt you, but I think it's better if someone like raises their hand up and so
yes.
to answer this question because it's sort of like an interview style question. I'll give you practice as well for next year or like in a few months when you're applying.
So
So
if
any
anyone
brave volunteers raise their hand up.
go on. I'll unmute and then you can unmute and then speak. Please introduce yourself as well. The person with the hand up. I don't want to try butcher your name.
Oh, sorry, I did not know if you were like referring to me or someone else.
Worries.
So I'm Equa, and I think one of the biggest sort of things you'd have to consider when you're giving a person such a diagnosis is HIV and AIDS is something that's been thrown around quite a lot within whether that be the news or what other people are saying. So I think the public perception is quite important. So yeah.
Real. Yeah, I completely agree. So, with that stigma, and I suppose if someone feels like that diagnosis would be attached with stigma, what kind of skill, there's a big one I'm thinking of, should a doctor use when they approach to give this kind of big news?
I think quite a few would probably say like you have to be direct or something, but I think what's the most important here is to have a lot of empathy and to be very sort of sensitive when you're saying like such information.
Brilliant, yeah. Okay, now let's say this is someone who's come in and they are in a committed relationship with someone else. What is your duty as a doctor in that situation, bearing in mind thinking about how HIV is transmitted? It's up to you if you want to continue to answer, Equa, or someone else can also put their hand up.
Oh, I didn't know. Sorry, I didn't know if someone else wanted to answer. I'm like happy to answer if you want me to.
Yeah. So confidential. There's two in the chat. We'll just go from the chat. I think that's more than fine. So confidentiality,
Okay.
but also looking at the risk of harm as well to other people. And so yeah, Al, are you happy to move on? Anything to add?
Yeah, I think we covered everything. Maybe that empathy is a quality, but the skill you show is communication. So how do you show empathy is by communicating, sort of actively listening to what they have to say, making sure that the patient feels heard. So that distinction is a bit important.
Yeah. Okay, so the next kind of transmission is mother to child. So this is through three ways. So in utero, so via the blood, just across the placenta. And this is the main way. the virus is in the blood and in the infected cells of the mother, goes across to the child, and the child then acquires the disease as well. However, there's also some limited evidence that it might be via the amniotic fluid and membranes which surround the child. And then also during childbirth, so maternal fluids and blood are ingested by the newborn, leading to infection. So as I said, you have mucosal tissue everywhere. When the child ingests it, the virus then goes across the mucosal tissue in the gut of the child. And then breastfeeding as well, similar, crosses the gut mucosal tissue. However, how HIV reaches the breast milk is not yet known. There's a few different hypotheses for that. Okay. And then the last main way is percutaneous injection. So this can be through blood transfusions, vaccines, and a main way in a few developed countries as well, especially in some areas of America, is drug use, which is administered like intravenously. So that's again just the HIV just goes straight into your blood and then can cause infections from there. Okay, so another interview style question. Again, please, I'm going to say try not to put anything in the chat while someone's answering. I'd much prefer someone volunteers and gives a spoken answer. How can we reduce the transmission of diseases via percutaneous injection? No one? Oh, Khan. Okay.
Hi.
Hello.
I'm Zara, and I think that maybe you could reduce it by having like specified bins and stuff for needles,
Brilliant.
because otherwise people leave their needles on the ground, and then young children can pick them up, and obviously that can spread it.
Brilliant, yeah. So that's kind of like a very isolated incident. Very good answer, very possible, very viable. But what about in like a wider, I suppose, a healthcare context, thinking about
Raise awareness, like through campaigns, maybe have like public figures sort of talk about the risks of sharing needles and percutaneous.
brilliant, yeah. How about in thinking about blood transfusions, how are the main way that we've stopped HIV being transmitted through those in developed countries? I might hand that over to Shirazad if they want to give that a go.
Yeah, thank you. I think in lots of developed countries they test any blood that's being transfused for different infections like HIV and other infections that can be transmitted via the blood. And also just to add on to the other point of like drug needle injected related infection, I know in some like developed countries like Canada, I think they have specified like needle exchange programs where you bring in your old needle and they make sure that gets disposed properly and they give you a new one to make sure new infections aren't being passed on.
Brilliant, yeah, spot on. Yeah, and testing as well is really important, and also pre-screening as well. Before you donate blood, you obviously have to do like a questionnaire, and then see if you're at risk of carrying any disease or any viruses. So brilliant. Ali, do you have anything to add?
No, I think you guys gave a very good answer between all of you.
I agree, yeah.
Very well done.
You're really good. Well done. Okay, let's move. Okay, so now we're going to get into some more of the sciencey stuff. So firstly, from A-level right now, you probably know that there's these attachment proteins, which are divided. Now we're going to divide them into two kinds. You've got your surface ones and your transmembrane ones, your GP120 and your GP41. And they're in the envelope category. And then you've got these GAG ones, which are all your structural proteins. So the matrix, the capsid, and some other ones which surround the RNA. And budding protein, which will be involved in how the HIV leaves the cell. You've also got the genome RNA, that's what the G stands for, VPR, which is one of the accessory proteins, and then POL, which are the different enzymes which contribute to how HIV is replicated, and then the membrane, which is from a host cell's phospholipid bilayer. So the tropism of a virus is what cells it infects in your body. So as you guys said earlier, the T helper cells is the main one, but it infects any cell which expresses the CD4 protein, which acts as a receptor for a protein on the HIV virus, one of the attachment proteins. So they have a complementary shape. And it also can infect, though, macrophages and monocytes, which are types of phagocytes, and as we said earlier, dendritic cells, which are a type of antigen-presenting cell. So the strain that the HIV is, so it can either be R5X4 or R5X4, so it can be both. And this is determined by a hypervariable region, which means there's a loop of protein, a loop of polypeptide, and the sequence of amino acids can be very different between them. And it's a V3 loop on the GP protein. So it's kind of like a protein. It's got this polypeptide loop hanging out. And this loop determines which secondary receptor, called a chemokine receptor, is used to enter the cell. So the R5 ones are focused more on macrophages, and they're used to establish an initial reservoir HIV cells, because a phagocyte can then progress to be an antigen-presenting cell, as hopefully you know from A-level biology. And the X4 virus strain uses another receptor, which is more present on T cells, which progresses the infection forward into AIDS. Okay, so firstly, I'll go into a little bit of detail on how that process actually happens. If I can get my notes back up. Please hold. So firstly, what binds is the GP120, which I mentioned earlier, which is your attachment protein, onto the CD4 receptor, and it forms a complex with it. And then this will engage and moves the associated co-receptor, so either your CCR5 or your CXCR4 along with it. And then this will allow a fusion peptide, which is your to move into the cell membrane, which allows your transmembrane protein to open a gap in the cell membrane, allowing the capsid to enter the cell. So the capsid is the protein with the associated RNA and your enzymes inside, and also the matrix as well of proteins.
Oops, sorry.
Okay, so after the capsid enters, it moves along microtubules, which are sequences of proteins. A lot of proteins in this that act as your cytoskeleton, which allow things to move around the cell. And it uses something called actin, and myosin together, which you will learn more about in year 13. However, not in the context of the cytoskeleton. And it then moves the capsid to the cell, to the nucleus from the cell membrane. While this is happening, the process of reverse transcription happens, which, as Ali said earlier, is how we get from RNA to DNA. So during this process, however, I think I'll go back to the slide in a second as well, but what you can see here is something called A3G exists inside our cell. So our cells have small polypeptides and proteins as well, which are designed to protect us against viruses. And in normal viruses, they can then mutate the DNA. So here we've got a C swapping to a U in the RNA of the virus. What kind of mutation is that? Let me see if I can get the chat back open. If we're swapping a base for another. Substitution. Brilliant, yeah. So this substitution mutation can lead to a few problems. So it means that the RNA can't be integrated, the DNA, that's produced from the virus can't be integrated. And also, it can make a premature stop codon. So do any of you know from bio what a stop codon is? Not quite stops replication, but ends the sequence, kind of. Stops the making of mRNA, tells enzyme to stop replication. So kind of. So in a stop codon, you have a terminates protein synthesis, pretty much, yeah. So it's at the end of a sequence of RNA. You have a sequence of 3 bases, and it basically tells the ribosome, stop making the protein, just release the protein now, it's done. So basically, if you have this stop codon earlier, the protein isn't done making, and then you just have this shorter protein which isn't functional. So we end up with just non-functional proteins. So the virus can't continue to replicate. Also, it can also deaminate the RNA, which stops reverse transcriptase being able to work, which we'll go into more detail now. So, reverse transcriptase is kind of said in ALO biology. It's not really just like, it's just you don't really go into the process, but we will now. So, first thing I want you to bear in mind before I go through it is you have in the viral RNA, you have 2 sequences which are the same at either end. So to start the process of the reverse transcription, a tRNA primer binds at the start, and it produces a short DNA strand. So bear in mind for this, the green is RNA and the purple is DNA that's being produced. And it produces a short little strand at the start. Now, because it's complementary to the other end as well, it basically jumps across and it moves to the other side. And this allows a DNA strand to be produced across. And after this, after you've got the first DNA strand here at D, the RNA, most of the RNA is removed using RNase. Just another enzyme removes it. And here we end up with 2 primers left, which then continue the process of DNA replication. And DNA runs anti-parallel, so then you end up with the second DNA strand. And at the end, you're left with this triple-stranded structure, which is just called the DNA flap, the central DNA flap. That's what the C stands for, which basically tells the capsid to break down and helps the RNA go into the nucleus. Does that all make sense so far? If anyone has any questions, I'll stop quickly now. If anyone wants me to go through that again, or any parts. Can you go through it again? I might do it at the end. I don't think I can go through all of it again, but yeah, because that might take a long time, but I can definitely go through it again at the end. Okay, next slide. So, after this, like I just said, the capsid breaks down. So and it forms what you call the pre-integration complex, which is the DNA, there's the flap again, surrounded by all these different proteins, which allow it to be imported. And using these proteins, it can hijack the cell's systems already to get into through the nuclear pore and into the nucleus. So this is how integrase works and how we actually get the RNA into the viral DNA. So here's the viral DNA with the integrase on it. It's called a multimer. That just basically means there's multiple proteins there, like a quaternary structure. And the first step that the integrase does is makes these hydroxyl ends, so an OH group. Why do you think it'll make a hydroxyl group at the end? Thinking about your biomolecules. and those reactions. Not quite hydrolysis, because we're not making something. We are joining two things. It's a condensation reaction. Brilliant. So because we are joining this DNA strand onto the host DNA, these hydroxyl ends allow that. And then we have something called a provirus, which is where the viral strand of DNA is into the genome of the cell's DNA. Okay, so now the DNA of the virus is in the cell's DNA, we can then start to make the HIV particles. So another A-level bio question. What kind of microscope was probably used to produce this image of the production of the HIV? So someone said TEM, an electron microscope, transmission electron. Brilliant. Can someone give me a reason why? 2D, brilliant. And it's also in black and white, but they might be colored and stuff post-transfer. So black and white and 2D. And you can see inside, not just the surface. Very nice. And internal structure, brilliant. Perfect. Okay. So, as we can see here, the black is the protein from the virus, and it's budding at the surface, and then it gets cleaved once it pushes past the membrane, and then you end up with a HIV particle. Another thing as well that none of you mentioned, magnification. Viral particles are very small, so to be able to produce an image like that would not be possible with a light one, because it would need a very high magnification. Okay, as we can see, when it's integrated, translation happens, we get the viral structural proteins, but we also get these regulatory proteins, which we'll go into. Here we've just got a little bit more detail about how it actually integrates. Again, HIV and viruses in general do a lot of hacking what's already in the cell. So these two are proteins which attach to the end of the viral RNA to begin assembling it, which then allows these proteins down here to cut away at the cell membrane and allow it to be released. Okay. So one last thing as well, could you just go back one? I just forgot to mention something. The proteins here are just assembled in a lattice right now, and the proteins are just not assembled into the capsid and stuff, and the RNA assembled like this. So this is where the proteases come in to help form the overall protein structures. Okay, so I'll go through this when I can. Just now. Oh yeah. So we have 6 main ones. I think I'll just go through. I'm gonna go through 2 of them. So I'd like you to look down here first. We have something called TAT, which is part of the HIV, and it recruits something from the cell called PTFB, which allows it to activate RNA polymerase. And this means it acts as something called a transcription factor, which you will do in year 13. And a transcriptional factor is a protein which then proceeds to activate a RNA polymerase and means a gene is expressed. Another quite interesting one as well is the NEF one. It stops CD4 on the surface being taken by lysosomes. So, I mean, it does mean CD4 is taken by lysosomes so that the cell can't communicate with the immune system and be killed. And teverin as well. Teverin is a protein which acts as a signal to other cells that it's infected with a virus, and this is stopped by VPU. And yeah, I think I'll just go through those ones for now. Rev helps the RNA of the genome be released from the nucleus. So yeah, those are the different things that HIV does. And in summary, HIV wants to turn this cell into a factory which lives for ages and just makes loads of HIV. It stops doing its normal functions and just makes loads and loads and loads of HIV particles. And I know that was a lot of science, but in summary, when you are infected with it, it begins to attack all your CD4 cells. And over time, the amount of HIV increases. and the amount of T cells and other CD4 cells decreases until you get AIDS. And when there is less than 200 cells per milliliter, that's when it is defined as AIDS. And the main way that HIV proliferates and continues to replicate is we want the immune system to be constantly activated. We want these CD4 plus cells to live for as long as possible. So during that chronic stage, the latency stage, the infected T helper cells continue to live and produce those HIV particles, and this is why we need the immune system to be activated. So this may be brought on by co-infection. So if you're infected with hepatitis B and Epstein-Barr virus, HIV damages, again, as I said earlier, the mucosal layer. And if this is in the gut, you might have heard of the gut microbiome. So bacteria from that can enter your blood and then continue to activate the immune system. Interferons are basically just also things which attack the immune system, which HIV can raise the levels of. Obviously, the replication of it before it begins to hack the cell can activate the immune system as well. And all this can cause a lot of inflammation, which can lead to other chronic diseases during that time and increases the risk. So even with effective treatments, even if it just stops HIV before it progresses to AIDS, it can still lead to increasing risks of lots of different very common diseases, or which other risk factors, which there might already be other risk factors for as well. So yeah, I think that was the second to last slide, or might be the last one on my side of it.
Oh, sorry. I think my wife just. Are we back?
Yeah.
Okay, so well, you're done with the other slide, right?
Yeah.
Okay, so can we get a volunteer to describe this graph, learning like all the science behind it? Now, like sort of, can you describe this graph?
Some tips I'll give before you discuss it. So I don't really know about other MMI universities. None of the ones, I think Imperial might as well. You might be given a graph. So with this, start from basics. Just explain what you know about the axes, what types of axes they are, and then just describe the shape of the graph. And when you get to a point in it where you know there might be a possible explanation for it, you can stop and explain that. If you don't, just keep going. So yeah, if anyone would like to put their hand up and volunteer to have a go at that.
Even if I feel like even if you don't understand it completely, just raise your hand and like sort of speak through it, and we'll guide you as well. So yeah, Khan, you can sort of open your mic and start speaking if you're ready.
Yeah, I've no idea what's going on. I'll just try. So on the x-axis, it seems to be showing the time period. So from weeks, and then it's got this sort of gap, and then it goes 2 years. So I'm assuming that means that like there's the biggest changes that occur at the start. And then the y-axis is the CD4 plus lymphocytes. So that's the type of cell which was the CD4 plus, and it's showing its concentration, I assume. And then that's correlating with the blue line, whereas for the red, it says HIV RNA copies per milliliter. So that's a concentration of that. And then for the blue line, it has a big sort of dip, and then it goes up a little bit, and then it goes down at like a constant gradient. Whereas for the red line, it goes up and it peaks at around like 7 weeks. And then it goes down quite a lot until 9 weeks, and then it's pretty flat and it slowly increases, and then it increases quite a lot from 10 weeks and beyond, and then that leads to death. So I'm assuming that means that a high HIV RNA copies could be an indication of death. I mean, first time, not necessarily, but the second time, yes. And then it's also seems to be split into three categories with initially acute HIV syndrome, and then in the middle period, clinical latency, and then at the end, AIDS symptoms.
Brilliant. Okay, I think we'll stop you there. That was really good though, and literally that's exactly what we meant. Just start from the basics. Say, okay, what this is on the x-axis, this is on the y-axis. I'll just ask you one quick question. What is the difference between how this axis is encountered and how this axis is? Looking at possibly.
Powers, I guess. So like with the left axis, it's the numbers are much lower, so it only goes up to 1,200. Whereas on the right axis, the numbers are going up by powers of 10.
Brilliant. Do you know what kind of axis that's called?
So,
It's absolutely fine.
no.
It's called a logarithmic one. Any of you might have heard of that.
Oh, yes.
I think Heba said it in the chat.
Brilliant.
But yeah, that was a very good answer. Like literally everything I
agree
put on it was exactly the same. If like sort of ideal answer would be exactly what you said. So you sort of break it down, talk about the axis first, and then talk about the trends, and then explain the trends. So say for example, this is clinical latency. This is obviously in a real interview, they would probably not give you these you can see my cursor, right? They probably wouldn't give you these labels, and they'd ask you, okay, so tell me what you know about HIV. When do you think AIDS would start? So stuff like that, right? So that's what they'd be trying to sort of get from you, see what knowledge you already know. So yeah, I think that's it for pathophysiology, and now on to treatment. So detecting HIV is very important, and the way it's detected is that we use ELISA tests. I think you learn about that in AQA. It's basically you test if a protein is present. So you can either test for antibodies or antigens to see if they're present in the blood of a patient. Third generation ELISA test is just like a different. Obviously, we in AQA we learn first generation sandwich ELISA test, maybe stuff like indirect, but there's obviously it gets more complicated than that. But these are good for these are good for detecting an immune response after it already happened. So after the virus has already replicated itself, after your body has initiated this immune response. So these only show a positive test result after 2 to 3 weeks after the initial infection. So another test that would be better is nucleic acid test, so PCR. You do this in AQ as well in year 13. And it just detects it detects if there is HIV RNA in your body, and you can be monitored, which is the next slide. You can be monitored your viral load, which is how much HIV RNA is in your body, and how much CD4 lymphocytes you have. So how much how much white blood cells you have. That's basically the graph that we got Zara to that's the graph we got Zara to explain, which was showing the CD4 lymphocyte count go down as well as the viral load going up. And the goal for viral load is we're trying to get it to be undetectable, because that means it's untransmittable. So if it's below a certain level, then it's undetectable and untransmittable. So that's the goal with treatment. That's what we're trying to do when we give patients antiretroviral therapies, is to get the viral load to be undetectable again. So when the doctors test, they test when there's a risky exposure. So like we said, with the needle injury, unprotected sex, if your mother is HIV positive and she just gave childbirth to you, so you'd be tested as well. And if you have symptoms or opportunistic diseases appear, so for example, if you have TB, tuberculosis, you'd be tested for HIV to see what sort of course of action should be taken, because a lot of the drugs used for tuberculosis interfere with the drugs used for HIV. And there's also routine screening for example, like we said before, blood donors, STI clinics, and just in general. So antiretroviral therapies, these are the gold standard for HIV patients, and the main sort of the main way that they used is in highly active antiretroviral therapies, which is what HAART stands for. These are basically a combination of many different classes of antiretroviral therapies. So each class targets a different stage of the viral replication cycle, which I've put here. This is a summary of it. And each stage sort of targets a different each class targets a different stage of the cycle to stop it or to inhibit it. So when we use 3 different classes, we're basically preventing HIV replication in 3 different phases, which limits how much the disease can progress and slows down the progression and increases that sort of clinical latency period. So the first of these drug classes is entry fusion inhibitors. These work by stopping the HIV from entering the host cell. And one example of this is ibalizumab, which binds to the CD4 receptor and causes a conformational change. A conformational change is basically a fancy word for changes the tertiary structure. So it changes the tertiary structure of HIV GP120, which was one of the attachment proteins, and it doesn't change the structure of that. It changes the structure of the attachment of the CD4, which means that this attachment protein can't bind to it, but it still allows for MHC class 2, which is basically, if you did Edexcel biology, you'd know that this is what causes B cells to be activated when helper T cells bind to them and form this complex, so multi-histocompatibility complex. So that means that immune function is still preserved, but the HIV is blocked from binding to it. And then similarly, you've got Maraviroc, and it blocks the CCR5 protein, so HIV can't enter the cell. So these are the proteins I'm talking about. So CCR5, the HIV needs to bind to both the CD4 and the CCR5 to be able to fuse with the membrane and enter the cell. So if we block one of them, that means that the HIV is unable to enter the cell. And for fusion inhibitors, if I tell you that they act as a competitive inhibitor to P41, which is one of the envelope proteins, how do you think that works? So how do competitive inhibitors work? You can type it on the chat or open your mic. Yeah, so they bind to the active site, and that means that there's less of the enzymes needed to fuse or like break down the envelope are able to do so. So that means that no fusion occurs, or like limited fusion occurs. Another class of drugs is reverse transcriptase inhibitors. So like we said, reverse transcriptase turning RNA into DNA to be incorporated. And there's 2 different types of these. There's nucleoside and nucleotide. And they both work by interrupting the translation process by ending the replication early. So like we said with the stop codon, right? Imagine the stop codon was in the middle of the viral RNA. So that means that the DNA getting made, it doesn't get made because the reverse transcriptase stops in the middle of it. So you effectively stop this sort of section. And then you've got integrase inhibitors and protease inhibitors. So integrase and protease, we said integrase causes the integration of the HIV DNA into the genome of the host cell. So if we stop this, then we basically stop the proteins that are required for HIV to replicate from being made. And same with protease inhibitors, they stop the sort of formation and assembly of mature HIV viruses. So like this should be on this arrow actually. I forgot to change that. But yeah, this should be on this arrow because you need the protease to sort of release a mature HIV particle. other treatments that an HIV infected patient might take are prophylactic treatments. So these are, wait, actually, this is not for infected. This is to prevent them from being infected. So if someone's at risk, for example, someone who's sharing needles, they would take prophylactic treatments to ensure that they are protected against HIV. HIV and tuberculosis, when they're together, like we said, they provide a particular challenge because the drugs interfere with each other. So we need to make sure that we use effective sort of timing and make sure to time the antiretroviral therapies in a way that it doesn't interfere with the tuberculosis drugs. So we've got another interview style question. So like we said, prophylactic treatment is there to prevent HIV from developing. So do these type of treatment, the preventative treatments, do they benefit the NHS or are they burden to the cost? So I want you guys to like sort of go through both sides and let me know. If someone raises their hand, that would be
Even if you just want to give 1 point, that's fine and explain it. You don't have to give like a full answer.
yeah, actually, go ahead.
Could you say that preventative treatments would be beneficial to the NHS because by preventing, in this case, HIV, it would reduce sort of the number of cases of HIV, which so I'm not sure about the costs, but I'm assuming the preventative treatment would be less expensive than the use of antiretroviral drugs and sort of the treatment after someone gets HIV. So in the long run, it would be more economical for the NHS as well.
Yeah, that's a very good point, and especially because HIV is a chronic illness, so it stays on for a really long time. So preventing it and starting off from early will save a lot of money for the NHS. But I'm really glad you said long term because obviously in the short term they need to invest a lot in the prophylactic treatments. So yeah. Shahrazad, do you have any other points?
Yeah, just to like discuss the other side as well. If as like a strict prescription criteria isn't like administered to like GPs or pharmacies, it might lead to overprescribing in situations where they aren't necessary, which provides a greater like financial burden on the NHS. So having like a really strict sort of narrow criteria that the person needs to fit before the drugs are prescribed could be beneficial.
And another thing to add to that as well, sort of because we said HIV is highly susceptible to mutations. What do you guys think if we give everybody these treatments? What do you guys think would happen? Yes, was it Equa?
Equa, yeah.
Equa, yeah.
So it would kind of end up being like a flu vaccine for the previous year. Like it has the risk of being ineffective because I don't know of maybe antigenic variation or something like that.
Yeah, exactly. So we'd have resistance to the drug if it's used poorly or is misused. So another thing I have here is like
Oh,
sort of
Zion, I think.
Zaun. What do you have to say?
I just thought it would be similar to like the like whole like MRSA like situation. Like people would become more they'd be able to fight it. Like they'd develop like not as big a response to it anymore. And so it wouldn't become effective like the more people you give it to.
Yeah, so the more people you give it to, exactly, that means that the virus mutates more and then it gets like sort of with natural selection. Obviously, viruses aren't living, but they still sort of experience that. That the virus is resistant to that drug now. Another thing is that you can argue that it might not be allocated ethically, so sort of it's not gonna be given around equally. Not everyone will have equal access to this treatment, so it won't be as prevalent. Do you have anything to add? Halima, do you have anything to say?
I just want to say about the cost-wise. I think it would be beneficial to prevent it, like invest in drugs to prevent AIDS, only because if we think, yeah, it's expensive like short term, but long term, because how AIDS works is like you become more, the immune system weakens, so you become more like you can easily get other diseases that will require treatment. So in the long term, like getting those treatments for it, it will like add up, the cost will add up, and it might be just more expensive than just investing in like preventing AIDS in the first place.
Yeah, exactly. That was a really good answer. Everyone gave really good answers. Yeah, so I think I'll move on because of time, and we have quite a few slides left. But if you guys want to go back and add any points, that's fine too. So current HIV research is focused on many different sort of fields. So I want to explore those with you guys. There's been a lot of work on HIV vaccine development. So but it's been difficult because, like we said, HIV has has a really high mutation rate and genetic diversity, and it's able to integrate into the host DNA. And obviously, we don't want to harm the whole, like the person's lymphocytes or white blood cells. But there have been trials that have shown a vaccine efficacy of above 30%, which is a very it's a proof that a vaccine may be possible. And current research is focusing on broadly neutralizing antibodies, which can target conserved regions of HIV, of the HIV envelope protein. Other new approaches include mRNA-based vaccines inspired by the COVID-19 vaccine, and mosaic vaccines designed to protect against many HIV strains at once. So having a vaccine that targets all the different strains. While no licensed HIV vaccine exists yet, advances in immunology and biotechnology continue to bring the field closer to a breakthrough. So gene editing can be used to counter HIV, and there's been a lot of sort of discussion about this, especially with CRISPR-Cas9 and like sort of gene editing becoming a thing. This can be done because you can basically edit the gene for the CCR5 protein here and make it so that the cells do not express this. So that means that HIV can't bind to it and it cannot fuse with the white blood cells. So this is done by CRISPR, right? So CRISPR stands for clustered regularly interspaced short palindromic repeats. And this basically scientists saw that plasmids in some bacteria had these sections of DNA which just repeat themselves, and in between them would be viral DNA. And what they found out was that this is the bacteria's way for self-defense against viruses that attack them. So viruses that attack bacteria are called bacteriophages. So scientists also found out that this protein here called Cas9, it binds to those plasmids and it basically cuts any viral genetic material if it encounters it. So they basically took this idea and reused it for I'm doing something. And they basically used it for sorry, that was my sister. They used it for gene editing because in humans we have two natural DNA repair mechanisms, non-homologous end joining and homology-directed repair. So you can basically cause a double strand break. You can break the genome at one point and then it can be fixed by these methods, which will cause a change. This was actually done by He Jiankui in China in 2018, and it was done on an embryo. This is very controversial because this was never done before on embryos. Gene editing has only been done on stem cells for sort of therapeutic purposes, and it hasn't been done on embryos because the consequences aren't known yet, because obviously this change will be carried on in generations, as the whole embryo's body is based on that. Sorry, 1 second. Alhassan, can you talk about the next the
I'm sorry, I was on mute. So with genomic editing, there's also some ethical things to consider. Complications may occur. Obviously, if we end up cutting out the wrong gene, that can cause some pretty nasty complications. As a result, like with the gene editing discussed, and also if something in the body doesn't go completely wrong. Again, with any of these treatments, including and preventative measures, socioeconomic divide, if how they are treated. The research into CRISPR-Cas9 is very expensive, so we don't know how long we'll have until we have a treatment that can be provided cheaply. And something to consider as well, medical ethics. So when we're talking about this, we want to think about how will continued research into things like CRISPR-Cas9 what will that lead to in the future with following gene editing, things that may happen. Ali, do you have anything to add? I didn't really.
Yes. No, that was okay. Oh, is my Wi-Fi gone? Am I back?
Wi-Fi's yeah, I'm back.
Oh, yeah. My sister just needed help, but yeah. Anyway, yeah, that was very good. I heard what you're saying. Epidemiology, we'll go over this quickly. There's only like 4 slides. So statistics, these are some statistics on like how HIV is sort of how many people are living with HIV. But more importantly, I want to highlight sort of the progress that we've made. So in 1996, that's when highly active antiretrovirals were sort of produced and developed. So you can see that there's a sharp decline in the number of deaths caused by HIV after that. I want to actually ask you guys, why do you think that there was 0 cases of HIV before HIV didn't exist? Or
Perfect. Yeah.
yeah. So yeah, HIV wasn't identified until 1981. So obviously there would be no cases recorded until then. So I also want to highlight the inequality in sort of what areas experience HIV. And while HIV treatments have transformed survival in wealthier countries, there's still disparities, especially in sort of African countries, where co-infection rate is high. So they have a high rate of tuberculosis and high lack of access to antiretroviral therapies. And the combination of these things leads to a higher death rate from HIV. So this is a timeline of HIV. So like we said, the first report on AIDS was in 1981, and it was linked with HIV in 1984. And the FDA approved the first antiretroviral therapy in 1987. And in 1996, the combination treatment, which is what highly active antiretroviral therapies were developed. And in 2011, it was proven that antiretroviral therapies can reduce the risk of transmission by 96%. In 2018, the gene edited twins were born who were resistant to they were resistant to HIV, and now research is still ongoing, and the goal of treating HIV can solve one of the scientific mysteries, but it's been like a lot of people are working on it through vaccine development, genomics, and stem cell research. And if you guys want, there's some extra reading and other questions to explore in EPQ or anything like that. But yeah, thank you for listening.
I'd strongly recommend this one, by the way, especially for people who aren't applying to Oxbridge and stuff. These two are great. I've not read this one. There is a movie though, which I might watch. But this one, My Own Country, is a really, really good one. These are both set in America, but this one talks a lot about how the empathy and stuff, and working as a doctor, especially in a smaller deprived town in America, and the skills he had to use there. And again, there's these questions down here as well, which you can explore.
But yeah, guys, another point is like we learned, or like we were able to sort of learn all the stuff in the interview course that we did with MyUCAT, and me and Alsan actually met through that, and we became like really close friends. So I really recommend you guys apply to the interview course, or like try to join it, because it's definitely like a very good investment. And it's not just that you get like very good preparations for interview, but you also get uh you get a lot of like sort of support, not just through the tutors, but also through the friends you get to make along the way.
Yeah, I think it's compared to the UCAT program's obviously amazing. I taught on it this year. I don't think either me and Ali did it. But the community on the interview program is much nicer, and it's a really good program. It's so comprehensive. Like you cover basically anything that you could be asked about. And on that as well, you're not just told about it, but you're also given the skills to be able to answer very unfamiliar things as well, which is really important. So yeah, I'd 100% recommend the interview program to sign up for it when that's available.
Yeah.
So yeah, I think that's us pretty much done on anything that we'd want to cover. So if there's any question, firstly, thank you for sitting through all that. I hope you enjoyed it. But yeah, if you have any questions or anything, feel free to pop them in the chat and we'll do our best to answer them.
So sorry to interrupt, guys. Really important talk, really informative from Ali and Al Hassan. So thank you so much to them for putting on such an amazing event. But just wanted to kind of flag up a couple of you asking when the interview program will be open for sign-ups. Just thinking about, obviously, we don't have a confirmed date yet. So would be really great if you guys could all follow our Instagram to keep up to date with any updates on sort of sign up announcements and also the announcements of all these other events. So I saw a question about does this happen routinely? It happens hopefully most Wednesdays and Sundays. So keep an eye out on the Instagram for any updates on those events. So Instagram is up here, TikTok is up here, and then UCAT.me is our website. You can see all the different links for all the different things associated with us as well. And just a little bit more about the interview program. Alan Al Hassan mentioned that they actually were on that last year. Obviously, they're going to Oxford now, so super successful. We actually had a 99% success rate last year as well. So any unis you want, we will have a mentor there for you to help you through. And it's super interactive. As you can see, you can make some really good friends on it as well. And a lot of information there. So obviously here, it was super detailed in terms of like biomedical stuff, lots and lots of detail on kind of the mechanisms behind HIV. So for anyone feeling a little bit overwhelmed, please do not worry. You definitely don't need all of that information for your interviews, but it's super insightful to kind of think about the mechanisms behind certain diseases and things, especially for Oxbridge or any more scientific based universities you might be applying to, as well as thinking about maybe if you can link this to anything you saw in work experience, that would also be super good. So that was our interview program. We also do interview one-to-ones, and this is going to be with a tutor who is currently at or got an offer from the university that your interview is for. And this can literally be anything you want. If you want a combination of learning, so kind of talking you through hot topics and current affairs, or things like practice questions, mock style questions, we can go through literally anything you want. So that is basically some of the stuff we offer for interviews alongside these talks that happen every Wednesday and Sunday. So thank you so much for attending. If Al Hassan and Ali want to take any questions, we can definitely do that now as well. Anyone have any questions to finish off, or we'll kind of wrap it up here for today?
Yeah, and obviously if you have any questions that you think about after or anything else you want to go, you guys said you were going to go through some. Yeah, the reverse transcription stuff again.
Do you need me to share my screen again?
Yes, I'll stop sharing. Of course.
I can quickly go through that again. Slightly simpler.
I need you to tell me what slide it was.
I've got it open now, so I can quickly.
Oh, are you going to share your screen?
No, I'll just tell you what slide it is, unless you want to try find it. It was that
This
one.
one. I think it was this one.
Yeah. So if I can write, there we go.
Sorry.
Okay. So the first thing that happens is that you need to bear in mind is there's these two strands at the start and the end, which are very similar, especially the R1. The R1 is identical, and then the U1 at the 3' end and the 5' end are slightly different, but still very similar and complementary. So when this tRNA primer, so a primer is something that when it binds, it initiates the transcription off into the DNA. So the DNA is produced at this end first, but obviously that's not the whole length of the DNA. It's just made me lose the ability to write. Oh, it's back. So you can then, there's a little piece of DNA at the end, which then moves, and it acts as a primer for the first strand, and it allows the DNA to be produced, a strand of DNA to be produced. And then the RNA is removed, except for two strands, two in the center, which then act as primers again and allow the second strand of DNA to be produced. And this also leaves a flap, which is used for signaling to continue the progression of the HIV replication and for it to enter the nucleus, if that makes more sense. If anyone has any further questions on that or anything else.
If you guys also have any questions about like applying or the interview program, or me and Al Hassan both applied to Imperial and got offers as well. So if you're not thinking about Oxbridge, you can also ask us about.
How did scientists discover this process? That's a very interesting question. So one that you could definitely look up, but from what I know, is things like just hypothesis and then testing it through investigations. There's some very handy imaging techniques, but there's definitely people.
Yeah, sorry to interrupt you, but also like the sort of I don't know, we did in AQA biology, we did this the semi-conservative DNA replication theory and sort of the Nielsen, oh wait, what was it? Messelson-Stall experiment. So sort of those types of experiments to see how well your hypothesis holds.
Should you include science in the detail of your personal statement? So I think the advice is, and it'd be really useful if someone with a bit more experience fact checks this. I think for Oxbridge, it's quite important. You want to have something quite sciencey that you've done on there. For other universities, you want maybe a little bit just to say, oh yeah, I have done something. I will be interested in the medical sciences if you let me onto the course. But it's not like completely necessary. And for more work experience based universities, for example, Sunderland, that space might be better for talking more about work experience from what I know about the personal statement. But that I might not be 100% correct. But I definitely know about Oxbridge. It's extremely important to discuss that.
Equa has the hand up.
Hi, thank you. So you see, if you're like interested in, maybe it's not necessarily like an illness or something, just like an insight like this, for example, like I really caught on to like the inequality aspect, like around the world, because I'm someone who's had like insights into medicine, not only from like the UK perspective, but also from like the Ghanaian perspective and how it's like there. So an insight like this, would you mention this in an interview, or is this more like a personal statement type?
I think you mentioned it both. I think that's a very important insight that so you need to talk about it's not just the disparities like sort of internationally, but also within the UK as well. So the discrimination within sort of like BAME communities having less access to healthcare, and not just BAME communities, but like just think about wider sort of like age divide, so all these different gaps that exist in our society. That's definitely if you're applying Oxbridge or even any, I think most unis as well, they could ask you about stuff like that. But yeah, it's definitely a very valid point that you can sort of apply to many different sort of questions that you may be asked in an interview. And I think you should also like sort of try to mention that you're someone who believes in sort of justice, which is the fourth pillar of medical ethics, and like that
Mhm.
the access to resources should be equal.
And this is all covered very well in the interview program. I remember that lesson. Yeah. So yeah, it's extremely important. I agree with everything Ali said. It's very important to discuss in your personal statement and interviews if that's something you're interested in. So yeah.
I'm doing IB
Thank
and we do
you.
IA. So IA is like an essay they do for every subject. Do an IA to do with
With
the
titration.
method.
Oh, that's what an IA is.
For me, for example, don't know how impressive that is. Oh, that's not a question.
Oh, well, when I was reading something, I think it was either last night or the night before, titrations are actually used sometimes to like measure viral counts in labs. I think, no, it wasn't for HIV, it was the book I'm reading on my reading list about coronavirus. They use titration to measure viral counts when they're like testing for vaccines and stuff. So if you're doing like, maybe if you're not doing Oxbridge, it might not be very important, but if you are, that could be quite a
I
cool thing you could try.
I talked about it in many interviews actually. I did titrations with my like sort of with a group in my school as part of like A-level chemistry, but I didn't say it was part of it. But then I used that for like teamwork. That I was able to work with people to achieve a goal and sort of to fulfill the task that we were given. So you can definitely spin it in like a different way. Or they do it alone.
Prepare for your interview alongside.
Prepare for your interview alongside the interview program.
To
So
be
the
honest,
yeah,
do you want to
do
take it?
answer this one?
Oh, I'll answer this one. Go on.
I-
How did you prepare for your interview alongside the interview program for Oxbridge and non-Oxbridge unis? Ali, would you say you didn't really have to do that much?
I think they set us up for it. Like when they knew someone's interview was coming up, they'd be like they have a like we'd share resources and stuff. We even had the first lessons without the tutor like that we set up
Mhm.
in Al Hassan and other sort of students. But yeah.
Yeah, I think compared to UCAT, obviously with the UCAT program, you do need to put in a lot of your own practice and your own time. But for interview, you can get away with a lot of the practice stuff yourself. I'm so scared for uni. Me too. But yeah, UCAS, honestly, I think compared to UCAT, with interview, you can kind of see the end goal a bit more, and you can really just delve into medicine and see why so much you enjoy it. So I think try not to be scared of the interview program, not interview program. Try not to be scared of the interviews, and I'd strongly recommend the interview program. But yeah, don't be. It's amazing. Yeah, I hear a lot of good things about uni and medical school though, so it's scary, but it's chill. And remember, even when you go to interview, they're just completely normal people as well. You can smile with them. I remember Ali messaging me after his first Oxford one saying I was cracking jokes with them. Like, they're normal people as well. They're nice. Like, it's not too bad. I think if we've not got any more, we can wrap it up there, I think. Yeah.
Yep, sounds good. So if nobody has, one more question.
Do you recommend not including everything in your personal statement if it's a uni that bases your interview around personal statement? So I'm not.
I'm not sure you can include everything in your personal statement anyway. There's kind of limited space. So I'd
Yeah.
say try and focus on a few of your examples that you can really flesh out to match that personal statement format. And then obviously all of the other stuff you've got to talk about, you can talk about that stuff in your interview that you've mentioned in your personal statement as well as everything else. So I wouldn't worry about kind of not having enough things to talk about because it's been mentioned in your personal statement. They'll ask you plenty of other questions aside from your personal statement as well, even if it's an interview that is based around it. So I wouldn't worry at all. Amazing. Anything else? Will the thresholds for UCAT be announced when data is released? Yes, the decels will be announced, the preliminary ones in I think, a few weeks, and then the actual final ones a bit later after that. Oh, for interview program. Yes. So hopefully once UCAT season is over and we've got all your scores, we will announce the threshold for that prize almost.
Trying to bargain.
Okay, amazing, guys. So anything else before we finish off? No? Okay, amazing. Thank you so much, everyone, for joining us. And we would love to see you on our future talks as well. We have some really, really interesting ones coming up. So please join us then as well. And hopefully you'll consider the interview program after seeing these two and the amazing friendship they have. Okay, so thank you, everyone. And we'll see you next time. Bye-bye.
Thank you very much.
Thank you.
Bye-bye.